When microalgae grow in nature, their relative success depends on many environmental factors, such as sunlight, temperature, availability of nutrients, access to adequate water or soil, the interaction with other organisms, and many others. And a lot of times, these changing environmental conditions are far from ideal, and this limits the growth of the microalgae. And in contrast, when we have intensive cultivation of microalgae, there is a combination of knowledge and technology, and this process takes place under carefully controlled conditions in specially designed photobioreactors, restricting any contamination and allowing us to have a fully controlled and optimized growth of the microalgae. The cultivation protocols are established to maximize oxygenic photosynthesis, which is a complex biological process that was actually developed by microalgae around 3 billion years ago, and it completely changed our world forever. And depending on the end use of the biomass, the parameters are defined and the protocols are established for each of the strains of interest. And these protocols include values for pH, temperature, CO2 consumption, the oxygen evolution, the concentration of critical nutrients in the medium, among many others. It is also possible to eliminate or introduce certain stresses so that the microalgae can react and adapt to them and create specific cell forms or compounds of interest. And all of this control allows us to have a guaranteed quality, constancy, and stability of the microalgal biomass. In nature, microalgae can be found basically any habitat you can think of, from aquatic environments or soil, even in snow, hot springs, or salt lakes. And in these natural environments, microalgae appear as part of a consortia with other organisms, like other microalgae, bacteria, fungi, among others. In contrast, in intensive cultivation of microalgae, microalgae are grown in axenic cultures, which means there is only one species per culture. And this ensures that you have the desired species, that there is an adequate biomass composition, since all the growth parameters are adjusted to optimize the biomass yield and quality of that particular species. To have axenic cultures, it is essential that the initial inoculum contains only one single species, and that all the operations related to the culture are performed under extreme caution to avoid any contamination throughout the cultivation process. Microalgae cultivation starts with a single cell, and it can be scaled up to billions of cells and millions of liters of culture with a very high cell density. The scaling up process begins with microalgae stocks of all the different species of interest, and these stocks serve to inoculate a very small volume of liquid media that contains the nutrients that the microalgae need to grow. This small volume of liquid media that's been inoculated is incubated under controlled and predefined conditions that are established for each of the strains. The microalgae can adapt to these new conditions and grow, and the volume is progressively increased until enough volume is obtained to inoculate the desired photobioreactor to produce industrial volumes of microalgal biomass. So the scale-up process allows the generation of industrial volumes of biomass and also of high-value compounds, starting from a single cell that is maintained in solid medium. During intensive cultivation of microalgae, we can adjust the growth parameters in order to control the composition of the biomass with an enhanced production of high-value compounds. For example, pigments like astaxanthin, beta-carotene, phycocyanin, also particular fatty acids, and many other biomolecules. And this range of compounds is of great interest in many industries and has applications in agriculture, food, aquaculture, cosmetics, energy, nutraceuticals, pharmaceuticals, and many other industries. Virtually any compound that you can find in a plant, there is a microalga that can biosynthesize it. They are like tiny biological factories. And it is not in vain that microalgae are the origin of omega-3 and other polyunsaturated fatty acids in fish. They're also indirectly the reason why salmon is red, and they have one of the highest protein content in nature. Microalgae are photosynthetic organisms that efficiently fix CO2 from the atmosphere at a rate of almost 2 kilograms of CO2 per every kilogram of microalgae biomass that is produced. Altogether, they capture about 200 gigatons of CO2 per year, which is more than half of the world's annual carbon sequestration. And microalgae in the ocean, that are also known as phytoplankton, they drive a biological pump that moves carbon from the atmosphere into the ocean, bringing down the atmospheric levels of carbon dioxide, and thus playing a vital role in fighting climate change. In intensive cultivation of microalgae, we can use CO2 that is emitted from power plants or other industries, and this CO2 would otherwise contribute to the greenhouse effect. And this way it is possible to positively impact the environment by using the CO2 for microalgal growth.